Hello and welcome to my channel. Today's topic is structure of skeletal muscles. As we know that there are more than 600 muscles which are present in our body and they are responsible for producing movement at different joints. So in order to understand those activities, we must focus on the basics, that is structure of a skeletal muscle. So in the structure of a skeletal muscle, let me give you a brief idea regarding it, that muscle fiber or the muscle cell, these are the two terminologies which are used interchangeably. Second thing is, muscle fibers, they are arranged parallel to one another. Third thing is, they are multinucleated. And the last thing is, muscle fibers, they are long, ranging between 1 to 4 centimeters and cylindrical in shape. So these are the basic idea about the structure of the skeletal muscle. But still, there is a lot more things to see. So let's get started with our main topic. So here you can see in this diagram that this is the muscle and here it is attached with the bone with this kind of strong structure. This is known as a tendon portion. So tendon is the very cord-like structure and very strong in nature, and it is directly attached to the bone. Next thing is, this much area is known as a muscle belly. Muscle belly, or I can say the fleshy area of a muscle. So now this fleshy portion of a muscle is covered by a connective tissue sheath is known as an epimysium. Let me mention it over here, that this much area is covered by a fibrous sheath, fibrous connective tissue sheath is known as an epimysium. So inside the epimysium lies the fleshy portion of the muscle. Now if I take the transverse section, so here you can see multiple bundles are present. These bundles of the muscle fiber, they are covered by another connective tissue sheath named as an perimysium. So this area or this connective tissue sheath is known as an perimysium, and inside it again there are structure which is covered by the innermost layer is termed as an endomysium. So these are the fasciculi, or I can say the bundles, which are covered by the perimysium, and this particular area is covered, let me change the color. So this covering is known as an endomysium. So I can say the endomysium is the innermost covering, but actually a typical muscle fiber or the muscle cell, it is surrounded by a plasma membrane which is known as an sarcolemma. So as I have mentioned the shape of the muscle fiber, that these are the long and the cylindrical. So for example, if I draw a muscle cell, so you can imagine that this is a long as well as cylindrical in shape, and it is covered, let me draw a covering over here, and this is completely covered by a membrane which is known as a plasma membrane, or I can say the sarcolemma. And inside this typical cell, a cytoplasm is known as an sarcoplasm. Now actually the real theory portion starts, that when you get an idea about a muscle fiber or a muscle cell, that it has a typical covering known as an sarcolemma, and the cytoplasm of a skeletal muscle is known as an sarcoplasm. So this idea is now clear to us, that these are the two terminologies. Now we must understand one important thing, that sarcoplasm contains many things. So as I have mentioned earlier, that these muscle cells, they may contain one or more nuclei. They can be multinucleated. So the nuclei are present just beneath the sarcoplasm. just beneath the sarcoplasm, the nuclei of a muscle is present inside the sarcoplasm. Sarcoplasm also contains many other cellular organelles, that it contains the Golgi apparatus, it also contains ribosomes, endoplasmic reticulum, and the most important structure, that is myofibrils. Myofibrils are the most important because these are a vertical, or I can say the parallel lines inside the sarcoplasm. So when you cut this typical muscle fiber and you look in the transverse section, so you would find that this is the transverse section, and you would only find some of the dots which are present inside the sarcoplasm. So these dots are actually the myofibrils. And myofibrils, let me give you an idea of an electron microscopic structure of this myofibrils. That myofibrils, they show a typical characteristic that there are bands, that is A bands and the I bands. I hope you all are familiar with these terminologies. If not, I will explain that what are these A band and the I band. So let me draw a diagram of a myofibril. So in a myofibril, there are some A band in the centermost area, then comes I band, so the area which is black, so this typical area is the A band, and this are the portion of the I band. So why do we call it as a A band and the I band? So A band means it is anisotropic to polarized light, and the I band which is isotropic to the polarized light, which means that whenever we give a light rays or we strike the light rays over this myofibrils, what happens is from the I bands light rays are refracted at the same angle, but from the A band light rays are refracted at the different angles. So this is the reason that we name it differently, that A band means the anisotropic and the I band which means the isotropic to the polarized light. I hope now the identification of the A band and I band is clear to you, and to make it more easy, let me give you another idea that whenever a polarized light strikes to the A band, so it appears dark in the electron microscopy, and whenever a polarized light strikes to the I band, so it appears light in the electron microscopy. So appearance of the A band and I band, so depending on that we also call it as a dark band or the light band. So do not get confused between the A and I band, as well as another terminology, that is the dark band and the light band. In this dark and light bands, so you can see that the centermost area, that is the A band, that is now clear to you, A band, and the little bit periphery, that is I band. Now the centermost area, that is, as you can see in the middle portion of the A band, which is also known as an H zone, and in the center of the H zone there is one line which is named as an M line. And one more important thing that we must focus, that I band is divided into two portions by means of a Z line. So that is the idea about the A band, I band, H zone, which is the centermost area where there is absence of the I band, and in the center portion of the H zone there is M line. I band is divided into two, which is the outer portion and the inner portion, and which is divided by the Z line. So this was the idea about the electron microscopic picture of a myofibril. Now let's come to the next and the important concept, that is the sarcomere. Now sarcomere, we must understand this concept. So sarcomere is basically structural and the functional unit of a skeletal muscle. So sarcomere, do remember this thing, the area between the two Z lines. So Z line is exactly in the center of the I band. Let me draw it over here. Z line exactly in between. Z line divides the I band into two equal portions. Area between the two Z line, this much area, is known as an sarcomere. So area, remember again, repeat with me, area between two Z lines is known as an sarcomere, which is structural and the functional unit of a skeletal muscle. Now, what are the components of the sarcomere? So components of the sarcomere, which includes, first is the A band, second is H zone, third is M line, and the portion of the I band which is nearer to the A band is also included under the components of sarcomere. So this is the idea about the sarcomere. Next important thing is that there are some molecular level, so the proteins which are present in the skeletal muscles. So these all band, that is A band, I band, they are made up of the protein molecules, that is the actin and the myosin. So actually this is the out of scope of this video, and I am going to explain this thing in my next video. But do remember this thing, that the actin which is a protein, so actin, it is present in the I band, and the myosin which is present in the A band. Do remember this thing, I band is made up of the actin protein and the A band is made up of myosin. And myosin, it is a heavy molecular weight protein, whereas the actin is little bit less than the myosin. Now the last point in the structure of the skeletal muscle, that is the sarcotubular system. So let me clear all this thing. Sarcotubular system is very interesting. So we know that in the muscle fiber or the muscle cell, there are many cellular organelles are present, that is mitochondria, Golgi apparatus, and with all this there are two tubules which are present along with the myofibrils. So these two tubules, they are known as the T-tubules and the L-tubules. So let me draw a diagram over here so it will be clear to you. Let me draw it vertically so it will be easy for you to understand. So these are the A bands, outer area. These are the light band, or I can say the I band. Now for the T-tubules, as we name it as a T-tubules, which means these are the transverse tubules. Transverse tubules. So why do we call it as a transverse tubules? So the transverse tubules, they actually travel transversely through and through the muscles. So these are the tubules which are open from at the both of its ends, right? For example, this is a tubule which is transverse and which is open from one end and also open from the another end. And these are the tubules which travel through the muscle, and in that one end of the T-tubule which remains outside the muscle, and the another end reaches the maximum depth inside the muscle belly. So the function of the transverse tubule is that this transverse tubule helps to move the extracellular fluid inside the muscle cell, as well as whenever the action potential comes. So the travel or the transmission of the action potential impulses, it becomes very easy when a T-tubule is active and T-tubule penetrates the muscle through and through. So suppose this is the muscle, and the transverse tubules, they are traveling through and through. So whenever an action potential comes, so it becomes very easy for a myofibrils which are present exactly horizontal to these transverse tubules, very easy to achieve or to receive the action potential from the T-tubules. And T-tubules also help in the conduction of the extracellular fluids. Next thing is the L-tubules. L-tubules are known as a longitudinal tubules. L-tubules, or I can say the longitudinal tubules, they correspond to the endoplasmic reticulum of the cell. So endoplasmic reticulum of the cell, it forms a sac-like structure at the end portion. So at the end portion, this sac-like portion is known as a cisternae. Let me write it over here. This forms a cisternae. And this cisternae, actually it lies just by the side of the T-tubules. So let me draw a simple diagram over here, that here is the myofibril structure, here is the T-tubule which actually penetrates it, and here is the bulged portion, or I can say the sac-like structure of the cisternae, or I can say the L-tubules. So these are the longitudinal tubules. Similarly, here also a sac-like structure is present. So this is a T-tubule. Here is the cisternae. Here also a cisternae is present, which is a bulge portion of the L-tubule. So this triad, that is bulge portion of the one cisternae, one T-tubule and the another cisternae, this triad is known as an muscle T-triad. Muscle T-triad, which is shown in this figure. So it becomes very, very easy to understand. And the significance of this is that whenever the action potential comes, as I have already explained, that it becomes very, very easy to travel through the T-tubule and the action potential reaches to each and every myofibril. But whenever it comes, it releases the calcium ion which are present in the L-tubules, or I can say the bulge portion of the endoplasmic reticulum. And this calcium ions are released, they trigger the excitation contraction coupling theory, which is the topic of my next video. So this excitation contraction coupling is initiated by the release of the calcium ion, and release of calcium ion can be achieved when an action potential reaches up to the level of T-tubules. So I hope you understand the concept of the sarcotubular system. If you are having any doubt, do let me know in the comment section. I will try my best to solve it out. If you like my videos, do like, share and subscribe my channel. Don't forget to hit that bell icon so that you will receive notification each and every time I post my new video. Till then, keep learning, keep exploring. Do share these videos maximum so that one can understand the topic very easily. Thanks for watching the video till end. Thank you so much.